T-minus 1 minute.
Hello, everyone. I'm Dr. Constance Metters. I am an IPA serving as the MSI STEM Engagement Liaison at Kennedy Space Center in the Office of STEM and Europe. Today, we are here because we are celebrating
T-minus 30 seconds. We are go for launch. T-minus 10, 9, 8, 7, 6, 5, 4, 3, 2, 1.
Hello, everyone. I am Dr. Constance Metters, and I am an IPA serving as the MSI STEM Engagement Liaison at Kennedy Space Center in the Office of OSTEM and Europe. Today, you are attending our second Courageous Conversation. The inaugural Courageous Conversation was a Juneteenth celebration that took place the day before President Biden announced Juneteenth as a holiday. Today's celebration is a Black History Month celebration, and we are celebrating the University Research-UR1 team, a collaborative team of HBCU researchers to have their research fly on the International Space Station. UR1 started as a small merit project that was coming to an end. The project was picked up by the International Space Station as a collaborative test project to demonstrate and test the capability of HBCUs to have research on the International Space Station. This collaboration was approved by the Director of Education at the time, Mr. Leland Mebane, and also it was approved by the Director of the ISS Program Office, and that was Mr. Mike Seppardini. This was approved with full ISS funding. The NASA team that supported this project was Mr. Willie Williams, who was the project manager, and then also Dr. Pamela Dinkins, who was an engineering research scientist, and Ms. Misty Moore, who was in the education department. The launch was in 2014 on the SpaceX. The ISS research payload team is now known as the historical team. So now I would like for you all to actually meet the team. And we'll start by having you meet the individual teams members as they go around and introduce themselves. We'll start with Dr. Sanderson.
Hi, good morning, everybody. Where do I start, right? It is so historical and it's overwhelming at the same time. I'm Dr. Alamedu Sundaresan, Lalita, as they know me in NASA. I served as the PI for this historical International Space Station University Research-UR1 team, comprising of 5 HBCUs, Texas Southern, Prairie View A, Savannah State University, Tougaloo College, and Jarvis Christian College. And our research was about the effect of benzofuran derivatives on the immune system, both the normal immune system and cells where the immune system goes aberrant, to test the therapeutic effects and the beneficial effects of these benzofuran compounds in space on these particular immune cells. And I'm from Texas Southern, and Texas Southern is in Houston, close to JSC, so we have a close partnership with JSC, and we were able to lead the team to a successful historic flight as we continued our collaboration from the National Science and Technology Institute. I'm a professor of biology and the chair of the Department of Biology here, and I used to serve as NASA as the Signal Transduction Lab director. And I think I shall pass on the mic to Dr. Marriott from Savannah State.
Hello, everyone. I am Carla Sue Marriott, and currently I am the director for the Forensic Science program here at Roger Williams University in Rhode Island. Prior to joining Roger Williams, I was at Savannah State University for 15 wonderful years. And during that time at SSU was when I participated in the UR1 NASA project as a synthetic organic chemist. So I'm the chemist of the group. And I'll pass the mic on to Dr. Wilkins.
Good morning. My name is Richard Wilkins. I'm a professor and graduate coordinator at the Department of Electrical and Computer Engineering at Prairie View A&M University. I've been at Prairie View since 1993. Prairie View's in Texas, and so it was easy for us to collaborate on the project. And we had a strong background in space radiation measurements, and so we did the space radiation measurements for the project. Thank you. I will pass it now to Dr. Mao.
Good afternoon, and my name is Jinghee Mao, and currently I'm a professor of biology. I serve as the Dean of the Natural Sciences Division, and I represented Tougaloo College as a faculty research for this HBCU-UR1 research. My expertise is molecular biology, genomics, and the transcriptomics. Thank you. So now I pass on to Dr. Bhuyan.
Good morning and good afternoon. I am Dr. Shakhawat Bhuyan. I am from Jarvis Christian College, and you know that Jarvis Christian College is in Hawkins, Texas, close to Dallas and Houston area. And I am a cell and molecular biologist. I work in this UR1 team as a research investigator, and we worked together synthesize benzofuran to see the effect in the cellular level. And also, I am very proud to part of this great team, and we've successfully launched our sample to the International Space Station. Thank you so much.
All right, thank you, everyone. And now at this time, we would like to have our welcome. So we will have Dr. Leslie Fletcher actually give our welcome.
Hello and welcome. My name is Leslie Fletcher, and I'm the OSTEM director at Kennedy Space Center. And 2022 actually marks my 20th year here at Kennedy. It's my pleasure to welcome all the guests tuning in today to listen to this amazing team of researchers. I want to also thank the UR1 team for taking the time out of their schedule to educate us on the research that they have done. And a huge shout out to Constance Metters. She's been instrumental in the short time she's been on our team. She's assisting NASA to collaborate more effectively with MSIs. And this panel is just one great example of her work. Thank you all for turning in. And now I'll turn it back over to Dr. Metters.
Okay, thank you. And at this time now, we would like to know more about the actual team and their road to research. So what I'm going to do is I'm going to have each one of the team members tell us a little bit about how they actually ended up on this project, what they were doing prior to the UR1 project. And so we kind of call this the road to research. And so since Dr. Sundaresan was the PI, we're going to start with Dr. Sundaresan again, and she's going to tell us about her path to the UR1 team.
I think, okay. So the path is always not so smooth and not so rough either. And so we were lucky. We actually had been a team under the MUREP NSTI, which is the National Science and Technology Institute, from Ames Research Center. And we were part of this immunology project where Dr. Marriott was synthesizing derivatives of benzofuran, which is a chemical compound that has been extensively used antimicrobially prior, and also looking at, she was also working on cannabinoid receptors on the immune system as well. So the benzofuran derivatives, there were two of them, and we had tested them out in ground-based research with a microgravity culture analog, the rotating wall vessel. And we found, so we used normal immune cells, the lymphocytes, which are running through your blood right now as you're sitting down, and also cancer cells, which is basically the immune system gone wrong or gone completely awry, multiplying kind of crazily, if you will, right? So one of the compounds she gave us was called K-MEG, and it was called KM12. K-MEG was very, very useful in rallying up the troops of the lymphocytes and making them multiply to get a good immune response in our ground-based research. And KM12 was very successful in doing away with the cancer cells. So we had two compounds doing different things, but both were good for your body. So when we found that in our microgravity analogs that these compounds were having these effects, then as the project wound down, serendipitously, Dr. Denkins and Willie Williams approached us to see if we would like to try our luck out in real space through microgravity, and that's how it all began. Then it went through it was a rocky road, but finally we made it through the ISS office with the help of MUREP, with the help of Willie and Pam, to actually through my Safradini, to actually have a huge project that lasted us for about 3 years from 2013 on. Until then, I had been working with NASA, but moved to the university, and I was also an immunologist. I had flown shuttle before, and we were close to JSC, and having had some previous experience, I was chosen to be the principal investigator of this project, and I totally and humbly served as the principal investigator. So that's my road to research. I'm an immunologist by trade. So I'm quasi an expert on immune cells. So that's where my expertise is, on human lymphocytes. And I am going to now pass the mic on to chemist, Dr. Marriott.
Hello again, everybody. So my story really started on a little island in the Caribbean. So I'm from Jamaica originally. So I graduated from the University of the West Indies in Jamaica with a PhD in chemistry. I went on to do postdoctoral research fellowship at Clemson University with Professor John W. Huffman in the area of medicinal chemistry. So I was working on the synthesis of cannabinoids. So if everybody knows what cannabinoids are, they're extracts from the marijuana plant. We didn't actually work with the natural product. We synthesized everything synthetically, man-made in the lab. So we synthesized cannabinoids to target receptors selectively in the body for the treatment of cancer, modulate the immune system, etc. So there are a lot of potential for cannabinoids in terms of medicinal aspects. Moving on from Clemson, so I spent 4 years in Clemson. I moved on in 2006 to take a position as an assistant professor at Savannah State University in their chemistry program. And I started up their forensic science program as well while I was there. During my time there, I got an RO3 research grant from the National Institutes of Health, the National Institute on Drug Abuse. And during that time also, I was still doing some starting out my work with NASA, and I met Dr. Denkins, Dr. Pamela Denkins, and she was very instrumental in bringing the team together because she introduced me to the rest of the members that would soon to be on the UR1 research team. So that's basically my historical road to research and how I got joined up into the UR1 project. My role was as the chemist, and I synthesized, as Lalita mentioned just before me, compounds that are actually derivative of benzofuran carboxylic acids. They're carboxamides, and I synthesized them all in a research lab at Savannah State University with the help of my undergraduate researchers, Shakima Bowman and Daniel Mace. And I'll pass the mic on to Dr. Wilkins.
Well, thank you very much. I'm Richard Wilkins, and my background is in physics. Unlike most of the rest of the team, my background's in physics. I have a bachelor's degree in physics from the University of Pittsburgh, and I have a PhD in condensed matter physics from the University of Michigan. I came to Prairie View A&M as a research scientist. Prairie View has had a long relationship with NASA, studying space radiation environments, space radiation effects, space radiation shielding, space radiation dosimetry for a long period of time. And so just before the UR1 project, I was the director of the University Research Center Center for Radiation Engineering and Science for Space Exploration, and we collaborated with Johnson Space Center on a variety of topics having to do with space radiation and measuring space radiation. And at the time, it just so happened that we had a graduate student by the name of Janet Ruetis, who was doing an internship at Johnson Space Center with the Space Radiation Analysis Group under the direction of Dr. Ramona Gaza. And it was through that internship that Janet was able to work with the UR1 team and was able to work, be a liaison with us with the radiation group at Johnson Space Center. And it was in that way that we were able to get the flight certified radiation monitors that were used on the payload. In addition, we also had a research scientist, Brad Buddy Gursey, who is a world's expert in radiation dosimetry and had done a lot of work in the NASA radiation biology community, and was a help to our research as well. With that, I will send it on to Dr. Mao. Thank you.
Thank you, Dr. Wilkins. Okay, so this is my name is Jinghee Mao again. So my background is both in medicine and microbiology, and I received my BS degree and master's degree in medicine from Wuhan University in China. In earlier 90s, I then attended the University of Mississippi Medical Center, where I conducted research on molecular characterization of iridovirus that caused the amphibian die-off in America. So, and then in 1999, I received my PhD in microbiology. After 2 years postdoctoral training, and I joined the Tougaloo College as assistant professor. And Tougaloo College is a small liberal arts HBCU located outside of Jackson in Mississippi. Before joining the UR1 team, I had been supported to do the research as a junior faculty in this such a small HBCU. I was really fortunate, and through, even though our primary responsibility is teaching, so the support from biology department, division of natural science, and the college, so we can do research, and the trainee student also develop our own professional skills. So my research interest focused on gene expression and in the different diseases, such as I studied type 2 diabetes in the prostate cancer, especially from different ethnic group. And also I did some lipid profile study in prostate cancer, looking for different, the races like the Black population and the white population. And so those studies are supported through the HBCU-UR via NSF and also Mississippi INBRI and NIH. So I have been able to publish papers on the peer-reviewed journals and mentoring so many students in my research lab and even in my classroom. So my expertise in molecular biology and genomics and transcriptomics really brought me to this team by Dr. Wilson, who was a professor at Jackson State University. He reached Tougaloo College and talked to me whether or not I'm interested in doing the NASA research. And I was so happy, I agreed I wanted to be this team. So that's how I get into this wonderful team. Before the UR1, we are the team through the MIRAP and NSTI from 2007. So now I'm passing on this to Dr. Bouyan.
Okay, good afternoon again. I am Dr. Bouyan, Shakhawat Bouyan from Jarvis Christian College. Just prior to, involved in this project, I was in the Jarvis Christian College. I just started my career as an assistant professor since 2006 at the Jarvis Christian College. And before that, I just worked as a postdoc in the UT Health Science Center. So if you just look, my background is cell molecular biologist, and I worked in this UR1 team as a cell molecular biologist. If you just remember the Dr. Marriott, so when she just synthesized the benzofuran derivative, there are two derivatives are here. One is the KM12 and one is the KMEG. So the KM12 has the anti-cancer effects. So our target to see the KM12, the benzofuran derivative, how actually kill the cancer cells. And also one of the idea came to our mind, we just hypothesized that if we use this KM12 in the cancer cells, and we can see any induction or induce the killing of the cancer cells in the space radiation. That actually came to our team, and then I am as a cell molecular biologist, and my prior experience, and also I started my the first NASA grant, what actually the NASA training grant with the UNCF SP, that actually we started in 2011 to 2000, I think that 2013. And also the team actually met with Dr. Pamela Jenkins, she actually collaborated and also bring all together in this team. So that actually the starting point. And then when we started like the project UR1, we plan to use the same the cancer cell, like the lymphoblastoid TK6 cells, and also in my part to see how actually these benzofuran derivatives can kill the cancer cells. So we actually started a lot of groundworks, a lot of dry run about this with this the KM12, and then finally, like 2014 in April, we actually sent this sample to the International Space Stations, and my part was to prepare the samples and everything. So that actually the real like the experience to work in the space radiation and also the experience to work as a group, so that actually and also the Jarvis students, they're also involved in this project. They work in the different, like the NASA Johnson Center, Kennedy Space Center, and also they presented their research in the different conference also. Thank you so much.
So thank you, everyone. Thanks to the team. And now at this point, we want to talk about the research. We want to hear about the research that was actually performed. We want to hear about your research experience with NASA and the International Space Station. And so now we're going to start the research part, with Dr. Sandra Reston, because she was the co-PI, and she's going to actually tell us about the research and what they did for the International Space Station. And so Dr. Sandra Reston, would you please turn your camera on? And let's make sure that everyone else, we all turn our cameras off.
I guess Dr. Bhuyan still has his camera on, but that's okay. That's
okay. Sorry.
Good, right? Things like that, fluxes. But anyways, I'd like to first start off by thanking everybody, especially Dr. Meters and Lester as well, who's been very instrumental in getting us all together again. I'm thrilled about it. And having served as a principal investigator, I'd like to first start off by saying that we all worked very well together, and our students worked very well together. That was a highlight, is to actually see these students' career points go from A to B. There were about 10 students in total. Before I start off on the research, I just want to acknowledge those students and how they were the whole fabric that knit us all together. And because they were constantly asking questions and keeping us sort of going, right? We were all we were not geezers yet, but we were getting there. So these students were the life of the team. So I just want to make sure. And TSU had 3 students, 1 undergrad and 2 graduate students who got their master's theses. And the research basically involved looking at the effects of KM12 and KMEG on cancer cells and normal immune cells, okay? And the benzofuran derivatives were synthesized, obviously, by Dr. Maria. We had already optimized a lot of the parameters in our ground-based research. We knew what kind of doses we wanted to use on these cells, but we had a long way to go before we said, okay, we can fly in space. So that's why we had a lot of dry runs. We had to optimize conditions to be able to fly. We had an incubator. It was called the CBA, which we shared with another investigator called Dr. Honglu Wu, who also had an experiment on the same SpaceX 3 payload. But have a way of getting carbon dioxide into the cells. We had to come up with that. And we'll discuss a little bit more about that when we had problems and those kinds of Q&As going on at that point in time. We had a dry run at JSC. We had another dry run for the radiation aspect at Loma Linda Laboratory in Loma Linda, California. So Loma Linda sounds like a nice Spanish word, but it's actually a very well-known university in California. So we went over there as well. And then we also had dry runs at KSC. We had to order blood. So where do I get these lymphocytes? And Dr. Meters, can you put up those wonderful cells? Let me show you what I'm talking about. If I could have this first slide. So these, we knew that lymphocytes were affected adversely in space. So these lymphocytes, if you look on the left, 1G is Earth gravity, right? And micro G is actually, it should be micro G on the other side, that's space. So what happens, that's right in the middle. So when you launch the cells, if you look at 1G cells, these are lymphocytes. These are isolated from your peripheral blood that goes in your blood vessels. We collect them from the blood bank. We run them through a series of steps, and then we isolate those lymphocytes. These are the guys that help you mount an immune response, guys. Without these, your immune response would not count, would not be effective. So they have to multiply first. When they go up to space, what happens in space? What is space? It is very close gravity to zero gravity, right? But it's not really totally zero gravity. ground-based systems we tested. So that's why it was important for us to fly to real space where it was true microgravity. In the ground-based vessel, we achieved close to zero gravity, but it's not quite the same thing, right? So in microgravity, if you look at the cell, what's happening to that cell is becoming rounded. It's becoming rigid. It can't function as well. So there's a lot of loss in properties of the cell, like the fluid distribution of the cell in yellow, gene expression. So obviously, everything changes when you take gravity from the equation. 9.8 billion years of having known gravity, right? Life on Earth. Once you take gravity away, things happen to even things as minute as cells. Lymphocytes don't move as well. They don't signal as well. So there's loss in signal transduction, locomotion, loss in their functional metabolism. They don't produce enough antibodies. They don't move well. They don't activate well. They don't talk to each other well. And they also, if you look at the rigid part of it, that's the cytoskeleton trying to adapt to the environment. It doesn't like microgravity, at least the lymphocytes don't. So they are becoming so round and G, which is on Earth, they're nice and fluid, they're so healthy, and they're ready to go gangbusters. And then when they return, it takes them a while. They don't automatically become all well again. It takes them a while to recover and become good cells again. So if you look at these lymphocytes, you see all of these changes in function, right? So how do you then make them multiply more? So that's where the KMEG came in. So we said, okay, if it's functioning well in the ground-based analog, will it do really well up in space? In the same way with KM12, it is killing cancer cells in the ground-based analog. Will it kill cells in space? And will it kill more? In that case, you know, do we have something that can boost the normal immune system and have good anti-cancer effects at the end of it, right? So these were some of the things we wanted to examine. If I can have the next slide, please. I'm just going to sort of walk you through the experimental Bob the Builder type situation. So if you look on the right at first, right, you have this rectangular box. That was the NanoRacks box we were given. And if you see each of these little strips of plastic, in that you have a tube that is separated in the middle by a clamp, okay? One side of the tube has cells with food in them, which is the media. On the other side is the fixative. So we actually trained the astronauts to open these up once they reach station on the 6th day so the cells were mixed with the benzofurans. So the lymphocytes were mixed with the KMEG. We had 5 replicates, and the cancer cells were mixed up with KM12. And on the 6th day, the astronaut opened the clamp by magic, and the cells and the fixatives were combined. Some of the cells were stored in -80, which is a freezer, to isolate RNA and look at the gene expression, look at the function. Remember in the previous slide, we talked about all those functions that were lost. So we can examine gene expression to see what happened to that. And then we also wanted to look at the anti-cancer properties, right? So some of them went to the freezer, some of them went to refrigerator to store till it came back after a month, okay? So SpaceX comes back after 1 month. So it stays up in station after the experiment is terminated, comes back up there. And inside these bubbles of plastic were basically polyethylene. You had carbon dioxide, 6% carbon dioxide that was diffused through the tubes and helped the cells live better. Because without carbon dioxide, they would die. And all of these were clamped together and put in that box, and it actually flew to the station. And it was kept in the CGBA, which is the incubator, okay? And the NanoRack module was there, and you can see one of those rams that are there, which was the radiation dosimeter. There's one inside the experiment and one outside to just measure the dose that's coming in, because radiation has effects on these cells. And radiation is synergistic with microgravity. That means it actually exacerbates the effects of microgravity when it comes to the effect on cells. So we wanted to separate those effects out. So SpaceX launched in April of 2014. It took about a couple of days to reach space station. And on the 6th day, the astronauts opened the tubes, and then they mixed the fixatives in the cells, stored it, and it came back. Okay, we'll talk a little bit more about how it came back, etc. We found that with the lymphocytes, and I have to talk about the lymphocytes, I'm going to let the others talk about their special aspects. But with the lymphocytes, we found that a lot of genes that were, well, we first found out that K-MEG was able to increase the population of the lymphocytes by about 75 to 83%, which means that it was able to recruit and mobilize the troops really well, activate those cells. And also, it was able to turn on the genes that enhance the survival of the lymphocytes. So that's why the functions were not as diminished as they thought they would be in microgravity when we compare to controls. And controls are also very important for these experiments because we'll have to separate the effects of what we're actually testing, okay? And we published 2 papers in microgravity and science and technology, and I can share the links with anyone that's interested. The students involved at Texas Southern were Olivia Madison, who is now a medical student at Meharry Medical College, Ivory Ellis, who's finished her PhD in molecular biology in Meharry, and I'm sorry, in what was it, Meharry or the other, I'll check on that later, but also Elvis Okoro, who finished his master's and now teaching elsewhere in Houston at University of Houston right now. So we had 3 wonderful students, and we also had a lot of undergrads. And previously with the NSTI, we had many undergrads who went to Johnson Space Center also to train. So with that, there is the patch. There is the patch. Dr. Marriott, do you want to go and then we talk about the patch?
Hi, yeah, sure, no problem. Do we have a slide with the chemistry?
So Dr. Barrett, you're just going to kind of speak a little bit about the K-MEG, and then we're going to move on. Thank you.
Okay, no problem. So pretty much our work at Savannah State was completed before we even went to the Space Center at Kennedy. So our chemistry was conducted completely at the lab, the research lab at Savannah State University, where the students, my 2 undergraduate students, Daniel and Shakima, would synthesize molecules, and we did the total synthesis of K-MEG took about 5 reaction steps. And what we would do, we would synthesize and then purify so that we're making sure that our final substance is completely pure, no byproducts or impurities. They would be purified either by recrystallization, which is simple, or we would have to do a flash purification on the column using an instrument, my favorite instrument, Biotage Isolera. Without it, we couldn't have synthesized so many compounds and purified them. So they were pretty much, we were very proud in our lab that when we synthesized compounds from our research group, they were 99% at least pure. So by the time we finished synthesizing the compounds, we would then take them down to Kennedy to pass on to the biologists in the group. And so that's when their work began. So that was a wonderful experience for us because it was like working with your own unique role and passing it off, and everybody working in synergistically. So it was very nice, especially the students that really enjoyed the experience. It was very holistic in that they got to experience every aspect of the research project, not only the chemistry side, but also the biology side, and they learned a little bit about radiation, etc. And then traveling to the Space Center was a very full experience for our students. Danielle and Shakima, Shakima is now a teacher. She always wanted to be a teacher, and that's what she is right now. Daniel went on to medical school. He always wanted to be a doctor, and that's what he is right now. Basically, that's it. I can pass the mic on to Dr. Wilkins.
Oh, thank you very much. As I said previously, our role in the UR1 project was to look at and measure the space radiation environment for the payload. And we actually did two things. We organized a what you would call a ground-based experiment to help prepare for the flight. As you can imagine, there's a lot of preparation that goes into one of these missions. And so one of the things that we did previous to the mission itself was conduct a ground-based experiment at the Loma Linda Medical Center, which is outside of Los Angeles. Loma Linda was one of the first facilities to operate a proton therapy for cancer, and the protons that are there, that are produced there, are similar to the types of protons, radiation protons that you might find in the International Space Station. And so we were able then to conduct an experiment that sort of in some way helped simulate the radiation environment that our payload would see when it was on the space station. The instruments that we used were something called radiation area monitors. This is a type of radiation detector that is called a thermoluminescent detector. And it's made up of material that when it's exposed to radiation, it will, the energy from the radiation will bump some of the electrons that are in the material to a higher energy level, and they get stuck at that higher energy level. And so they stay there. And later on, when you take the materials and you put them into a special heating machine, you can cause the electrons to go from that higher energy level down to a lower energy level. And in the process, they give off light. And you measure then the amount of light, basically how bright the light is that's given off, and that brightness of the light is proportional to the radiation dose that the RAM was, the radiation area monitor was exposed to. And so, as I said before, my graduate student, Janet Ruedas, was the real expert at these things. She had worked down at Johnson Space Center and had learned how to use the RAMs. She learned how to take measurements from the RAMs. And so it was she that performed all of the work and experience associated with this. And basically what we found when we got the RAMs from the payload, the UR1 payload, when we got them back from SpaceX, and Janet did her work, that our measurements showed that the radiation environment that our payload was exposed to was nominal. In other words, it was similar to what historical measurements on the space station had been over the up to that time that the mission took place. So, I will now turn it over to Dr. Mao. Oh, did you want me to say something about the patch?
So I would like for everyone at this point now to kind of turn their cameras on, and we're going to have a roundtable discussion. We're going to start with having some conversation about the patch, and Dr. Mao is going to start with us and tell us a little bit about her students and her experience, and then we'll move on with that. So let's talk about the UR1 patch, and we can start with Dr. Mao, if you want to give a little bit about the patch, please.
Oh, okay. Thank you. I think probably Dr. Watkins may be a better person to say this because it's really designed. And so I wanted Dr. Watkins to talk about, so he'll be more prouder than me
well,
to talk.
as I can't say enough about Janet Ruedas, and she really jumped in with both feet to this project. And in addition to doing the radiation work and working with the radiation group at Johnson Space Center, she was one of the folks working with the other groups that designed the patch that you see here. And she had input from all the various groups. Dr. Sanderisa's group, I believe, gave the DNA illustration here. And so she worked to combine all the contributions from all of the groups from the different HBCUs and combine them in this patch. And so if you notice the little stars beside the space station image there, each one is a different color to represent the different school colors of the HBCUs involved. And so Janet did that, and she was also very instrumental in keeping up with the Facebook pages and other media for the group. So she was a real leader, one of the real leaders of the group. Dr. Mao, do you want to say something about your research?
Yes, okay, thank you. Okay, so I think you have heard about the history about this research and also the chemicals and the radiation component. Now I want to talk a little bit about what my role in this research. So basically, like I said, my background in the molecular biology and genomics and transcriptomics. And so basically we studied the gene expression, differential gene expression in the cancer cells Dr. Sanderson mentioned about. So in this case, it's a leukemia. The cell we use is called TK6 cells. So this is a resemble the leukemia cell. So we wanted to look at how that chemical compound synthesized by Dr. Marriott can, to kill the cancer cells. Because from before this UR1 research we did, we have we spent a lot of time do the ground-based research, and then we checked actually both compounds, KMAC and K12, on the different cancer cell lines like skin cancer cell and melanoma, and also liver cell cancer cell lines, and also for the TK6 cell. So we use both compounds and turned out so there's a lot of the try out and determine the concentration, look at the killing dose and the comparison and the controls, and then we actually found out KM12 is a better candidate for the anti-cancer versus the KMAC. So Dr. Sanderson already described that one actually is more like immune modulation, but KM12 is like a cancer killing. So basically we see this, really they can kill the, the cancer cells to, really low proliferation. So that's why we choose the TK6 cells for the ISS, to fly the cells into the international space to see whether or not that KM12 can be, we know for the long-term flight, term flight, the astronauts have an increased risk for the leukemia, right? So hopefully this, from the ground-based research, we say they can preventing that cancer development. So we'll see how that, goes. So after we get a sample back from the International Space Center, so basically we have to extract the RNA to look at the gene expression under the environment, both microgravity and the radiation. And one group of cells without treatment of the KM12, another bag is treated with a KM12. So actually we found that a KM12 can really increase the sensitivity of the cancer cells to the what we call the oxidative stress. So we see more the reactive oxygen species expressed and less antioxidant species decrease. So from this study, we're really thinking the ISS, the setting for this kind of study really to give that promise for the KM12 as anti-cancer, the treatment. I think that's my role in this project. So now I'm turning on to Dr. Buan.
Okay, thank you, Dr. Mao. I am Dr. Buyan. I told you, so I am representing Jarvis Christian College, and I had like the 4 students actually working as a UR1 intern. One of like the Isla Dials, Cody, Terrence, Arkansas, and also Sandra Davis, all already they graduated, and they are now in the graduate program in different place. So I'm really happy, I'm proud with my students. They really worked hard. And as you know, my part was to grow the cancer cells that actually we say lymphoblastoid TK6 cells. That actually my lab, we grow and then we ready for the cells for the dry run and also the grow in, send the sample to the International Space Stations. So actually the research starting from my lab, and also I bring these cancer cells to the first, like for the dry run, to the Johnson Space Center, and also I took the samples to the Loma Linda. So one of the medical school, they have the opportunity to use the proton beam, the radiation. So our purpose was to use this radiation, and also I use the gamma radiation to see what actually happened in these, like the TK6 cells. So that all about actually prepare the cells for the International Space Stations. So finally, after Loma Linda, when we run or we simulate with the proton beam, already Dr. Wilkins also already talked about what actually we did. Finally, we actually sent the sample to the Kennedy Space Center. And Kennedy Space Center, we remember, like my students and also me stay almost a month just to grow the cells, to prepare the cells for the International Space Station. And the final day, we all loaded, and also we make the sample, grow the cells and put into the the nanorec, what actually we use to send the sample to the International Space Station. And my two students always with me to work in the lab, and they have also the very excellent opportunity to work in the NASA Johnson Space Center and also the Kennedy Space Center also, and also the Loma Linda. So I'm really feel proud the Jarvis students work with the cancer cells, and they also prepare the samples for the International Space Station. Then in the final day, I'm really proud to say my president, Dr. Newman, was present in that day of launch. So that actually proud of me, proud of Jarvis, and all encourage also me to be part of the program. And my president also encouraged my department, all actually encouraged me to be part of this. And also the day by day we improve all. And also when the samples came back from the International Space Station, actually my part was to get the TK6 cells, because this is actually the cells of the cancer cells, the mutated cells. And Dr. Sanders actually work with like the normal cells, normal lymphoblastoid cells, the lymphoid cells, like white blood cells. So that actually very clear the path, what we did, and also how synergistically we did the work. And also Dr. Mao, her part was to see the gene expression, but my part was to see the cell signaling pathway, how actually the cells are affecting or what actually the effect on the space radiation. So when we got the samples, we actually split the samples. Some samples I kept for my research, and also my students work with me, and some sample I sent to the Dr. Marriott and also Dr. Mao to see their part, and my part already I told you. So I see the signal transduction pathway, like the phosphorylation, and also how the space radiation, and also the ground-based research. Also, we have the control sample for the ground-based, and also we have the control sample, the space sample. So that actually we compare how actually the radiation can induce the killing of the cancer cell. That is a real, like the groundbreaking research, I can tell. Thank you.
Thank you, Dr. Bouillon. And at this point, I want to ask the team to turn on their cameras again, everyone. And I just want to say that this team, when I asked them about their research, they sent me so many slides and they were prepared to give these presentations. And every one of them could have given an hour-long presentation about their own research. So it's really difficult to condense it down into this presentation and combine this team. So I want to thank the team for trying to really pull their research in and to shorten it and to condense it. But I want us to move on because we've been talking about the research and we've talked about the students in the school and we want to talk about the impact. But I know that beyond all this hard work that you all were doing and all the work that the students were doing, that you were not just all work. I know that there were some fun things. I've heard some stories. The first time we came together, this team was just so excited, but there was one story that stood out and everybody kept talking about it. And so what I want to hear about is this. There's an alligator story, or I heard there was more than one alligator story. So it was somebody, I see Dr. Sanderessa, she's laughing, and somebody tell me about the alligator story. And then we're going to talk a little bit about the impacts and the outcome this had on your universities. And then we're going to open it for Q&A. And also want to make sure everyone knows that we had the chat available. So if you were typing in the chat, we'll address the chat. But let's hear about the alligator. Go ahead.
We cannot see the slide. We can't see the slide.
It's a big gator with its mouth wide open, Dr. Bouillon. So
yes, it is a big alligator with the mouth wide open. I'm sorry, Dr. Bouillon, if you can't see the slide.
Yeah.
That's what happens
but
when you start.
this gator was an 800-pounder. Guy was an 800-pounder. Yeah, this guy was an 800-pounder. And the previous day, what happened was there was another European experiment going on at another building called the Space Life Sciences building. So the I think foreign nationals weren't allowed on Kennedy. So one of my collaborators had an experiment. They had some trouble with the cell. So I went there around 6 o'clock after our work finished at Kennedy. And I was checking on the cells and telling them, do this, do that, and all of that. And they said, well, Dr. Sundaresan, you have some more time. You have a few hours to kill. There's a gator outside the door. You can't go home right now. You can't go back to your hotel. So it was another 3 hours before they let her out. This gator was sitting right outside the door. It's a big glass building. And it took them time to shoo it off. They didn't want to do anything, drug it or anything else. But the next day, they actually worked with it to isolate blood to see whether any environmental changes in the waters in Kennedy. Kennedy is on Merritt Island, which is a reserve, right? So every time on our way to work to get into, and that was the fun part, we would see gators, especially in April, along the streams, along the little, I guess, canals on the sides of the streets. We would see these guys all the time. Satro, the Kennedy Space Flight Center flight analyst who helped us, actually took some of us to see some gators later on as well. So, and the students just freaked out. We all went off from the lab to see the alligator with the blood being drawn, to see it getting caught and everything. And we got another call. Willie Williams is calling. So where are you guys? They said, we're going to launch soon. So come back. We got to prep the experiment. We raced back off again, but it gave us a moment of levity and the students really enjoyed it. So there are lots of pictures and photo ops. So the gator is the gator. Anybody else? Yeah, go on.
Thank you so much for speaking about the gator. So what I want to talk about now is I want us to talk about the impact of this research. And I want to hear about, from each one of you and a kind of a brief about the impact of this research on your career, on your institution, on your schools. What did this mean to have this type of research and to be part of history? And so we can start, we'll start with Dr. Marriott, if you want to start with the impact of the research.
Sure. Well, I think the biggest impact of the research was really in connecting what we did in our lab to the greater community, to the greater Savannah State University community, and to Savannah, the city of Savannah in general. I remember months after the launch, you could run into a history student that would be able to recognize us and tell us what we did in terms of the research, and that went into space. There was a watch party when the rocket was about to launch, which was in the library and other places. So students could actually watch the rocket launch into space. So that for me really raised research for me to another level, into not just doing research in a bubble for some topic that I chose, but to look at the bigger picture and to see how I can better serve the community, how I can leave something of impact behind. And so I think that's the real thing that happened for me during this project, is that instead of having a tunnel vision and a view of my research, I now sought to connect more with the community and people in general. And that actually moved me on to now focus, when I look for grant funding from the NIH and the NSF, I'm always having a student component, a research training component, and I've successfully gotten research training grants through the NSF and the NIH. One in particular that I'm proud of, that is still active at Savannah State right now, is the NIH at Desire. And it's kind of an integrative research experience, and it's to spark a desire for research with our undergraduate students, and with the added component that not to forget the community, to always have a focus on how your research can serve the community. And so I think that's what the NASA experience has really nurtured in me, community and interdisciplinary, integrative type experience.
Thank you, Dr. Marriott. So I know, Dr. Mao, you mentioned in one of the conversations about you all developing curriculum and how it impacted your institution. Can you tell us just a little bit about that?
Yes, and thank you, Constance. Yeah, I would, because first the 3 parts, I didn't mention any of my students, because I want to save the best for the last, right? Because yes, for this particular project, we have 2 students. One is Joshua Agee, another one is Amanda Osborne. So, and both are actually, so I grabbed those students from my microbiology class, and when I teach them, Amanda, actually, she was a social science major, but she tried to get a bachelor degree and then go to nursing. That's why she took my microbiology. And then when I, introduced my project, she got really interested. So she joined Joshua through her sophomore year. So both of them on the team completed those 2 or 3 years, the journey for this wonderful project. And after that, both of them, and now Joshua actually, he really loved the research, and he, from this NASA project, actually not those 2 students, I have 6 students and did a senior thesis and on the NASA project. And also I have 8 students working in the lab. And so those students are all very successful after they leave to Tuguru. There are 4 of them actually now the physicians, and Joshua just getting into the Meharry Medical School. He should be in the audience. He texted me, said, Dr. Ma, I'm going to Stanford to do the research as a medical student this summer. So this is a long-lasting impact on those students. Amanda, now she's a nursing. And also another thing is, during that period of time, I was able to create new courses. Actually, we called in the advanced immunology. So my whole lab session, actually we use that module, the microgravity apparatus. So we do the training 17 students in that class to do the NASA project to look at microgravity affect, innate immune functions. So start from literature review and project presentation. Students design their own research project, and then we do the hands-on experiences. After that, so we did presentation as a group, and they finally they write a report, the course report for that big project that lasts the whole semester. So, and this is another area also, I think really as a visibility as a small MSI, right? So Tugulu is such a small, so through that project, we are able to secure the other NASA grant, like NS, what I call the NASA App School. So I get that funding, around that period. Also, we get a small seed grant from the App School. And so as a small MSA, I compete with PWI. I think there's when I work with my collaborator at the University Mississippi Medical Center, they are so proud. They said, well, we don't even have a NASA project. You as a Tugulu representing Tugulu brought students to them. So we did some animal studies to look at, how that microgravity or radiation can cause the negative impact on the animal side. So this, I think, it really ended as a Tugulu, we feel like we so proud about as a member of this team, and so and other faculties through this project, other faculty in my division also applied a NASA grant, get funded to do the research. And what else I'm trying to say? Oh, also at one point we sent 10 scholars to the Johnson Space Center to do a summer internship. So I think those are my students see the value of the research, and so really prepare them to be a best physician or scientist in their career. That's what I wanted to share.
Thank you, Dr. Mao. So we're going to take about 1 minute for the rest of you all, if you please, to kind of tell just a little bit about it. And then we want to go into the Q&A. And then I would like for you maybe to give any maybe closing remarks or advice you have. But we definitely want to leave some time for the Q&A. So we can go ahead, if Dr. Buon, if you would just take about a minute to tell a little bit about your impact on your institution.
Thank you, Dr. Meaders. That actually a great effect, I can tell you, like the impact on the Jarvis and the Jarvis community. And you will be surprised to know that the time when we launched the sample to the International Space Station, so the media, the local media, newspaper, and also the TV, the KLTV, they just came to my lab and to see how actually the cancer cells, we spent to the International Space Station. So that actually the real like the story for the Jarvis community, and all like the local and also outside the Dallas, Houston area. Everybody knows that the Jarvis as a small school worked in the International Space Station. So that actually the great impact, and also my students, I told you, so they all are now in the graduate school. And also after impact, what I can tell you, so we got, I am as a PI, I got the one NSF grant, and also another one, I am the co-PI of this grant. That means the students, right after the NASA grant, we got the 2 grant from the National Science Foundation, and the students, every the year, the 5 to 10 students are working. And also one of the thing what I develop the undergraduate research as a course now every semester we are offering, and every semester at least the 10-15 students are enrolling, because they really know this is one of the path to be successful. To be go to the career in this, the biology or chemistry major. So that actually the real impact for this, the NASA project. So
thank you.
I just wanted to say something very briefly, just closing, I guess not closing remarks, but one of the things I'd like to be able to do, we were short on sample size for this experiment, so we were trying to get another UR2. So hopefully in future, we may be working together as a team, I'm hoping. And I also started to I also got another NSF grant, but also an NIH one and another NASA spaceflight grant with France. So it's an international spaceflight grant, and we haven't gotten a date yet, but we'll be flying at some point. But I also I'm going to get a machine that can do partial gravity, which means going to the moon and Mars. So we can try and experiment those kinds of cell paradigms as well, apart from microgravity. So that's something that we can think about in the future as well, to work together.
Thank you. So Dr. Wilkins, I know you didn't say anything on this section. Do you have anything you'd like to add about what happened at your university?
Well, what I would like to highlight is the fact that the project certainly did boost the image of Prairie View in our local community, and it continues to do so. Janet Roedes is a high school teacher in the Conroe Independent School District that's near Houston. And I spoke with her recently, and she says she uses her experience with the UR1 project all the time to inspire her students. And so I think that through her and through the other students, I'm sure that this project has had a very long-lasting, long-range impact on our community. Thank you.
Thank you. So one of the few last things I'm going to do is I wanted to share, and then you all received an award.
Yeah.
Not the alligator award, but you received a reward for your university research. And so I know that you have a copy of the reward, the award. Would you please read the award?
The NASA Johnson Space Center, the
Johnson
NASA
Space Center.
Achievement Award. Yeah, from the NASA.
Yeah, we got the National Aeronautics and Space Administration presents the JSC Group Achievement Award to the University Research-UR1 team for outstanding dedication and achievement in the development and conversion of ground-based university research to flight on the International Space Station. And it's individually to each of us, including the students. And it's signed on the 19th day of September 2014 by Ellen Ochoa, who was the director of JSC at that time. So that's the feather in the cap.
Thank you. And then I'm showing one last slide. And this slide I found was on a website, a Facebook page that was started by one of the students. And it says launch, and in the very corner of the slide, it says April 18, 2014. And so this was a slide or a picture of the actual launch. So I want to thank you all, thank the team. One thing I want to say is that we are really pleased to have you. It was an honor. It was an honor to meet you all. It's been an honor to work with you, to study and to learn about what you all were doing, what you're currently doing. And I want to go on and I want to say that we still have NASA research going on. We still have projects going on. Dr. Mal mentioned NASA EPSCoR and EPSCoR projects. We have the NASA EPSCoR FAST project, which is out there now. And actually this project was somewhat, the ISS was a forerunner to the EPSCoR ISS collaboration, of the establishment of that. So we have ISS projects and we have EPSCoR. So we have the National Science Foundation, EPSCoR/NASA FAST project that's out there. And we have other mirror projects. So I want to definitely encourage people as they are looking to engage and participate in NASA projects to please look at the projects that are out there. Please know that this team has opened themselves up. They have made themselves available to me, and they are willing to definitely be there for anyone else. They have experienced it. They know what it takes. And even though they make it sound really easy and they make it sound like it was just a lot of fun, we know it was a lot of work, but they've been willing to share that. And so with that, now I'm going to find out or see if we have any questions or anything that we need to address. And I think I will ask that of Lester. Do you know if we have any questions? So I'm not sure. I don't have access to the chat. Okay.
Yeah, thank you. Thank you so much. Thanks for joining with us. Thank you.
Well, thank you so much. Thanks everyone for joining us. And I want to thank the team for tolerating me because I know I have bugged you. I've called you a lot and I've texted you and I've emailed you a lot, but this has been an honor. And I look forward to maybe us doing something else. You mentioned your students, maybe doing something, a follow-up with the students and discussing the impact. And we're, yes.
I think it was 3.
Three.
Or 3 times. Yeah.
We had 3 scrubs.
Yeah, we started with the March 12th, and then we went again for like the April, and then finally we could, launch finally like the April 18th. So, yeah. Yeah, that.
Yes, we had to get the blood.
Yeah.
Our blood was 1 hour away from Orlando, so we had to isolate cells. We had to keep on. Dr. Buya had to culture the cells, freeze them back, get them back together, and then we had to reseal everything. And NanoRacks helped a lot, and I have to thank UNCFSP as well. They all pitched in, and there's a lot of prep. Yes, and you can reach out to us individually. We can help you out. We'll let you know.
But it's certainly the important thing that I think came about here was the fact that many of us had some sort of interaction with NASA before, and so then there were folks then in NASA itself that sort of knew how to assemble a team that that could work on a project like this. And I think they did a very good job. As has been mentioned before, the team worked very well together, and then we were able to do what we needed to do. It's a very complicated process to put something on the space station as it should be, and I think that with the help of the folks at NASA, we were then able to put this team together and then work together very well to make it successful.
Yeah. That actually is very important to sustainable. We started with UR1 and we had a very, impressive result, I can tell you, both in cellular and molecular, right? We see the space radiation actually can induce the killing of the cancer cells. That means if in future generation, if we think like the cancer patient, if we can send to the space, that maybe that way we can, treat the cancer patient also. So this is one of the thing, because our result actually showed this. And the reason we actually submit the second time UR2 project, what actually Dr. Sanderson mentioned, but still we are waiting. So then we may be better know about the condition, okay, cell in molecular level.
Thank you, team. Any other questions? Well, if nothing else, thank you, everyone. And I really want to thank the team, and I want to thank everyone that joined in. And I am, as I said, I'm super excited and I'm honored. I'm honored to meet the team. I'm honored to work with you. Thank you. Thank you very much.
Thank you.
Thank you so much.
Thank you for.
Bye-bye.
You're well. Yeah, thank you.
Stay safe.
Yeah, and Constance. Yeah. All right. So
Thank you, Lester.
Here.
Thank you. Bye-bye.
Thank you, Dr. Mendes. Thank you.
Bye-bye. Thank you.